These are the members of our groups, and this is the introduction to our experiment.
Step 1. We place 9 milliliters of distilled water into 5 test tubes. In order to do so, we use serological pipettes. For step 2, we first shake the solution to avoid any deposits. Then we take 1 milliliter of 0.5% starch solution blue in the tubes, and then we add it into the test tubes containing 9 milliliter of distilled water. After that, we mix the solution well and then leave it to settle. We label the particular test tube as 1. Step 3. We first mix the solutions, and then we take 1 milliliter of test tube A and then transfer it into the next test tube containing 9 milliliter of distilled water. We mix the solution well and then leave it to settle. We labeled is 2. Step 4. We take 1 milliliter of from test tube labeled 2 and then transfer it into the next test tube containing 9 milliliters of distilled water. We then mix the solution and then left it to settle, and then we labeled the test tube as number 3. Step 5. We take 1 milliliter from test tube 3 and then we transfer it into the next test tube containing 9 milliliters of distilled water. We mix the solution well and then left it to settle. We then label the test tube as number 4. Step 5. We will take 1 milliliter from test tube 4 and then we will transfer it into the next test tube containing 9 milliliters of distilled water. We then mix the solution and then we left it to settle. We label this test tube as number 5. Step 7. We heat the samples by taking them to a water bath of 80 degrees Celsius for 5 minutes. After 5 minutes, we then take them out and then add in iodine solution to observe the color changes from the samples.